Have you heard of stem cells, gene therapy, or cloning? If so, do you know what the controversy is surrounding these techniques? Where do stem cells come from? Is it possible to clone humans? You will learn these answers in this presentation. Stem cell therapy and cloning have ignited many ethical, religious, and political debates, which we will discuss more on slides coming up. Stem cells are unspecialized cells that can divide and differentiate into different cell types in the human body. Embryonic stem cells are found in embryos, and they can give rise to all cell types during embryological development. The controversy surrounding embryonic stem cells that are used in research and medicine is where the embryos come from. The embryos sometimes come from abortion clinics or are generated in a lab for the sole purpose of providing a source of stem cells. Adults do have some stem cells found in bone marrow and some other organs. However, these cells can develop only into a limited number of different cells, which makes them less desirable than embryonic stem cells. This may be overcome using relatively new technology called induced pluripotent stem cells. In this case, adult cells are reprogrammed to form stem cells by transferring certain genes into them. This would allow adult cells to be used instead of embryonic stem cells, removing the controversy. A developing embryo contains a large pool of stem cells that can give rise to all cell types in the human body. The cells can be removed and cultured to generate many cells to use for stem cell therapy to treat or cure diseases or injuries. Several diseases, including type 1 diabetes, Huntington disease, and Parkinson's disease, are caused by the death of certain cell types, making them good candidates for stem cell therapy. Type 1 diabetes is caused by damage to pancreas cells, while Huntington disease and Parkinson's disease are caused by malfunctioning nerve cells. Embryonic stem cells can be used to treat disorders or injuries. Adult stem cell therapy can be used as well, as follows. Stem cells can be removed from the bone marrow of a healthy donor. These stem cells can then be injected into a sick patient's bone marrow, where they should differentiate into normal cells, replacing the abnormal cells that are making the patient sick. An embryo can be generated in a lab to give a source of stem cells that can be grown and differentiated into different cell types to treat patients with different diseases or injuries. In adult stem cell therapy, cells are removed from a healthy bone marrow donor and grown in a dish. These healthy stem cells are injected into a patient, such as a leukemia patient who has cancerous white blood cells, where the stem cells differentiate into healthy white blood cells. Cloning is another application of DNA technology. Somatic cell nuclear transfer was used to clone Dolly the sheep in 1997 from an adult udder cell. Udders produce milk. Somatic is a non-gamete cell, so what this involves is taking a nucleus with the DNA from a non-gamete and putting it into a gamete. What this showed was that the nucleus from an adult cell can direct embryological development when transferred into an egg. While Dolly was born healthy and went on to have 6 offspring via normal sexual reproduction, she prematurely aged, having arthritis in her hind legs and dying of lung disease at age 6 when the normal lifespan is 11 to 12 years. It's thought that this is because she was cloned from adult DNA instead of fresh DNA from an embryo. Dolly the sheep was the first mammal to be cloned. To create Dolly, the nucleus was removed from a donor egg cell known as anucleation. The nucleus from an udder or mammary cell from a second sheep was then introduced into the empty egg cell, which was allowed to divide to the blastocyst stage of an embryo before being implanted into a surrogate mother. Dolly was born as a clone because she had the same exact DNA as the sheep on the right, from which the nucleus was taken from the mammary cell. Remember at the beginning of this lecture when I asked you if you thought it was possible to clone humans? Well, the answer is yes. If we can clone a sheep, cloning a human is really not much different. Somatic cell nuclear transfer or another method called embryo splitting can be used to clone any mammal, including humans. Potential benefits of cloning humans could be used to help infertile couples, or cloned human embryos could be used as stem cell sources, or they could be used to provide transplant organs. But the question remains, because we can clone humans, should we clone humans? There are many applications of DNA technology in medicine. It can be used to prevent, detect, or treat genetic disorders. Genetic disorders result from mutated, dysfunctional alleles. This means an allele that has changed and is no longer functioning properly. Dysfunctional alleles are identified or replaced in individuals to either diagnose or treat genetic disorders. DNA probes can be used to detect a disease-causing allele. A DNA probe is a single-stranded sequence of nucleotides complementary to a portion of the disease-causing allele. It's labeled with a fluorescent tag so it can be detected if it binds to a person's DNA. If it binds to the person's DNA, this means that they have the disease-causing allele. Preimplantation genetic diagnosis is used to detect disorders in embryos. For instance, if a couple carries recessive alleles for a genetic disorder, this method is used to screen for normal embryos. If two parents are heterozygous for a disease, what this means is that they don't have the disease, but they have a 25% chance of passing down two recessive alleles to their offspring and their offspring developing the disease. So in vitro fertilization, which is fertilization in a lab dish, is performed to generate an embryo that is checked for the presence of the genetic disorder allele. Therefore, a diagnosis of a genetic disorder can be made prior to implantation into the mother, which is why this is called preimplantation genetic diagnosis. Genetic testing is used to detect disease-causing alleles in newborns, children, or adults. The procedure is to take blood and extract DNA from the blood cells. Then PCR is used to amplify a potential disease allele to give enough DNA for sequencing to detect the presence of a mutation that would lead to a disease. For instance, if breast or ovarian cancer runs in a woman's family, she can undergo genetic testing to detect the presence of the BRCA1 or 2 genes, which would predispose her to developing one of these types of cancers at an earlier age. Gene therapy is used to replace a disease-causing allele with a normal one. For example, the cold virus, which is the rhinovirus, or adenovirus, which is another virus that causes respiratory illness, could be used to deliver normal CFTR alleles to the respiratory tract cells of individuals with cystic fibrosis. Cystic fibrosis is a lung disease, so viruses that specifically enter respiratory tract cells would be ideal candidates to deliver normal alleles. The problem with this procedure is that this is a temporary fix as the body's immune system removes the virus before all of the disease-causing alleles can be replaced with normal alleles. Gene therapy using an adenovirus vector can be used to deliver a normal copy of a gene to cure certain genetic diseases in which a person has a defective gene. While DNA technology holds many benefits for humanity, there are ethical issues to consider as well. For instance, should human genes be altered to treat or cure diseases, as is done in gene therapy, stem cell therapy, and embryo selection? While I think many humans believe that it's okay to treat or cure human diseases, there are those who think that any manipulation of human DNA should not be done. Another concern is that the human genome could potentially be modified for non-medical purposes, for instance, to select for better athletes, or for other desirable traits such as eye and hair color, or to treat non-life-threatening conditions such as baldness. After all, if DNA can be modified to treat human diseases, it can also be modified to select for certain desirable traits. As we leave the DNA technology unit, we consider many ethical issues. For instance, is there a good reason to clone humans? Should genetically modified crops be labeled so consumers can make a decision as to whether or not they want to eat these GMOs? Should genetic testing be done for disorders for which there's no treatment or cure? For instance, should testing be done for progressive and fatal disorders such as Huntington disease, for which there's no treatment or cure? Huntington disease is a neurodegenerative disease that usually has an onset when a person is in their 40s. Is it better for them to know that they're going to develop the disease but there's nothing that can be done about it? Or are they better off not knowing? The same question could be asked for newborns. Should newborns be screened for disorders for which there's no treatment or cure? Is this valid information to provide to parents that their newborns have a disease for which there's nothing that can be done? Other ethical issues to consider are as follows. How is the information in someone's genome protected from insurance companies, employers, and the government? For instance, if it's known that a person has a certain disease-causing allele, will this prevent insurance companies from offering them insurance, or will it lead to employers denying them a job? Should the results of genetic testing be shared with a person's family members if it may warn them about the potential to develop a disorder? Patient privacy laws typically make it such that physicians cannot share medical information with other individuals besides the patient. But is it unethical to hold back information from family members that could potentially save their lives? If we start altering genetics too much, could we start changing the evolution of our species? Evolution involves the changing of DNA over time, so if we start manipulating our own DNA, will we actually speed the events of evolution?